Welcome to Study Time, a televised home learning program produced by Rwanda Basic Education Board.
Hello viewers, welcome to the TV learning series, and here we are again for the Senior 5s. Remember, the unit is still Autotrophic Nutrition, Unit 7, according to the coverage of the syllabus, and we are still looking at how foods can be made. So remember what we looked at last time. We are looking at how light is trapped, then picked by the pigments, processed all the way through to get some organic compounds, some energy stores, some other molecules, release of the oxygen, but we have not yet got the glucose we want. So in today's lesson, we are looking at how finally can we have the food. What is needed in the light reactions that can be used as raw materials in addition to the gas, carbon dioxide, to have the glucose. That's the overall picture. But the question is, during these two forms of reactions, the light dependent and the light independent, is glucose the actual product of a light independent reaction? Or, so now that's something we are going to look at after. So let's focus on the slides and see what we do have. Objectives, one, like I said, remember, the topic is so sensitive in a way that we need to constantly link the previous lesson to the incoming lesson, the incoming lesson to the next lesson. So you do not want to take a risk of losing one part of it as a bit. So we shall therefore review the formation of the products we looked at in the light-dependent reactions, and then connect them to the light-independent reactions. Then we shall also look at how the carbon dioxide molecule, now something new that is coming up. We are looking at ATP, we are looking at NADPH2, we are looking at oxygen being released, we are looking at chlorophyll pigments, we are looking at light. Now this time around we shall head into the equation CO2 plus H2O. Now the CO2 comes in. How shall we fix it until we can finally have the product? So check, we do have carbon dioxide, we have water, we looked at that before, we looked at energy from the sun being trapped by the pigments, but releasing oxygen as well. Now this time around we are looking at how close are we to getting glucose as the product? The overall, at the end of the whole process, what does the plant have? So we are looking at reminding ourselves of what we have been going through in the previous lesson. So here is a quick go. Take it slow, but at the same time, not too slow, because remember, every second counts. So we do have the bilayer, a section of the membranes within the chloroplast. So we also have the photosystems we talked about, PS2, PS1. And then we also have the cytochrome system, a ladder form of arrangement. This is B6F. Then you also have what we call acceptor molecules of electrons. So this is plastoquinone, and then here you have ferredoxin. Now these, when we talk of the primary acceptors, these are the ones we look at. So let's check through. When light strikes a pigment, the electrons get excited, move to higher energy levels, and then pass through and received by a primary acceptor here, and then passed on through a cytochrome system. Guess where it lands? Where does it continue? We need to look at this arrow and check how it flows, whether it actually returns. That if we lose an electron here, does it come back? So if it doesn't, then that's what we look at as non-cyclic photophosphorylation. Photo, because light is used. Dilation, because ATP is produced. That's one. Two, that at the same time, water is split by photolysis, producing oxygen as the other product, waste, and hydrogen ions. The constant production of these is going to create an accumulation of the hydrogen ions here compared to this side up. So the amount is kept high here because of photolysis. With rapid diffusion, these will pass through ATP synthase, a proton pump now we do have. As these pass through to this side, we now do have ADP added to PI or an inorganic phosphate producing ATP. So we have phosphorylated ADP by adding PI, using the energy released by these protons as they get pumped into ATP synthase to form ATP. That's the sole role of light-dependent reactions, to be able to get this. But we also have one thing. As an electron flows through these systems, we refer to it as non-cyclic because it does not revert to where it came from. So it is lost through until it carries out the process of reduction, reducing NADP here to NADP H. So what are we talking about? Two main products of light reactions. One, NADP H, a reducing power needed in the next reaction, the light-independent reactions, and ATP as a store of energy for immediate use for these reactions. The enzymes controlling all these processes are going to require energy. So we say photophosphorylation because ATP is produced in the presence of light energy absorbed by the pigments, passed on until we finally have this. Now, with this, I want you to finally look at what we do have here. I want you to pay close attention and check this. You should be in position to check exactly what happens. How do we finally have glucose? What do we need? The materials, the energy being trapped by the green pigments until we finally have glucose. That in the light reactions, the product here we mainly have is oxygen. Glucose comes at the end. So what exactly happens? Solar energy, described in two forms. One, as a particle, a photon, but also has a wave motion. Remember, we're talking about wavelengths from here to here. So when we look at this, what exactly can we get? What is visible light? When we talk of the electromagnetic spectrum, what is usable by the plant? It's what we have here with a range of wavelengths. So when light strikes the pigments, as you can see, what is reflected and what is absorbed? We need to know which colors, which wavelength is important that is taken in, and which colors are reflected and bounce off. So they are not effective enough. The cell organelles, as we can remember in cell parts, all plant cells do have essential things, and one of them, the chloroplast, our store, our charcoal stove, our kitchen, among other parts. Remember, the membranes, the arrangement, and the design. If we look at these as the thyroid structures and how light, in terms of particles, photons striking with energy, reactions being divided into an area for light and an area for dark reaction. the light independent. But let's look exactly what we are looking at. The pump, the pigments, photosystems, and the electron carriers. Check exactly what is happening. Striking of light energy in photosystem. Here, how are the chlorophyll molecules behaving? How are the electrons getting excited and being released? What exactly happens in the reaction center? Bouncing off until we finally have enough energy to bounce these electrons off. Correct? Yes, there we go. Passed on through a system of electron carriers. But remember that at the same time, even this photosystem 1 is receiving the same energy. So this will release and this will release. So let's check exactly what happens. They leave here once excited and move here and passed on to the cytochrome system. At the same time, we lose electrons here. Water is being broken. So we have photolysis. Oxygen is released. And the electrons lost are replaced by what has come from water. These are passed on to a system as others do come. Now we have a problem happening. Every time water breaks down, hydrogen ions pile up here. Rapid diffusion as they get through here, the ATP synthase will take advantage of that and make the ATP as you've seen. So, low energy now passed on photosystem 1. Remember, they have moved from high to low and then handed over here. Now we need to check where these finally go. When we add electrons to a compound, we call that reduction. So what does this mean? Electrons are released, passed on to the BCF system, passed on for system 1, and then with low energy, the electrons reduce in a deep H as we form ATP, as you can check. Now, that's exactly what is happening. We have ATP and deep H as major products of the light reaction. Remember, this occurs in the thiacode structures being green, and so we can't have the next reaction taking place here. But before we go to that, what do we need to remind ourselves? Remember that the previous part we are looking at produced ATP and a deep H. The light-independent reactions. When we talk of the Calvin cycle, discovered by Calvin Benson, not me, take note of that, he designed a series of reactions by stopping one reaction after another and seeing from the time carbon dioxide is received to when we have glucose. What is the path? What is the stage? What is the route taken by this molecule of carbon dioxide until we finally have the glucose? Remember, carbon dioxide is one molecule with one carbon atom. But if we are to form glucose with 6 carbon atoms, the question should be, how many times do we need to receive a carbon dioxide molecule to be able to get the glucose? And if so, also, through what stages, through what components, by what enzymes shall we be able to trace that maybe after 1 minute, carbon dioxide molecule, which was 1 atom, is now 3. Then from 3 to 6. Or maybe from 3 to 5 to 6 finally, which we call a cycle. Because you see, received by 5, coming to 6, but during the course of these reactions, as we shall see, so don't worry about the small size, we have a much bigger one. What exactly happens? So he interrupted reactions, as we shall see in some of his experiments, at a certain point to check which product is in between. Bimeze nkaho wajya mu rugo ukatangira kwarura ibiryo bitarashya. After 5 minutes, you check the food. After 10 minutes, you check the food. After 15 minutes, you check the food. You'll be serving different foods between what you are cooking and what you're serving. So that way you're able to know that, okay, mu minota 5 ibirayi biba bimeze gute. Nyuma y'iminota 10 biraza kuba bimeze gute. Nyuma y'iminota 20 ndaza kwarura ready food, ready for consumption, iri different from ibyo nashize ku mbaho. You get my point now? So a cycle means that it's repetitive. Carbon dioxide has to constantly be brought in, fixed. But the question is, when you go for a wedding and you have these people muita protocole, I don't know how French translates into Kinyarwanda, but the protocol people are supposed to receive guests and pass them on to others who are going to direct them to where to sit. Do these people responsible for aligning people in their positions reach there and also sit, or they go back to the gate and receive other guests? So we have the receiver for carbon dioxide, hand it over, go back and receive more, hand it over, until we have all our 6 carbon atoms fixed into a glucose. And the process continues. So we need to constantly have carbon dioxide being received, carbon dioxide being fixed, carbon dioxide turning into glucose. Question: what if there is no light anymore? What about this? At the time when we have these formed, maybe within the first 5 minutes when light strikes the leaf, once they are finished, where shall extra come from? That's what we need to think about. That even when they are so separate, independent, because one takes place here where I have put it, and another one takes place here in the membranes. Are they interconnected? If yes, how? Which takes us to the objective, the link between the light-dependent products and the light-independent reactions. So it's like a bridge. I must have these if I'm to move to this stage. If I finish them, then this stage stops. Detaching the leaf before you can test whether light is responsible for this. Senior 1 experiments. So ATP and NADPH are reduced. NADPH is a reducing power, very heavy, reducing compound, produced during the light-dependent reactions, but will act as prerequisite, a condition needed before the dark reaction can take place. So check that out. These two are assimilated, used in the light-independent reaction. But why independent? Because we no longer need light, but we need its products, right? So that's exactly what we are looking at, the connection between the light-dependent reactions and the light-independent reactions. The products of one reaction will act as requirements for the preceding reaction. This area does not have oxygen because there are no chloroplasts where, there are no membranes where photolysis takes place. Possibly you also need to start thinking about, what if there was oxygen? Wouldn't these compounds most likely break down during oxidation and burning, simply respiration? Does it happen? What about situations where plants, during the day, even when there is light, they just decide not to have any foods being made? Like the rate of photosynthesis finally stops. What exactly are they trying to avoid? Could it be the accumulation of the oxygen we have got? So in one go, that the glucose 6-phosphate, which is a carbon compound, 6 atoms of carbon being produced, will be looked at in terms of what it is going to serve. 1, as starch for storage. 2, ATP. Starch, I repeat that. 2, cellulose. 3, glucose, maybe. That's why we're saying that all used for respiration. So when we say all used in respiration, we are thinking, is it possible that as soon as this glucose is produced, it is again used up by the plant? But for all that to happen, we must have 1, carbon dioxide as a second requirement, and 2, the ATP as another requirement, 3, NADPH, a reducing compound, to be used if we are to have the second and last part of photosynthesis, which is the light-independent reaction, taking place where? In the stroma. All we've been looking at has been in the thyroid membranes, photolysis, light reactions, photophosphorylation. Now this time here in the stroma, we don't need that. Instead, we need enzymes and ATP and NADP. No oxygen. That's why we hardly can find this region pigmented. Remember what we talked about, compartmentalization, division of labor. Reactions are not going to mix up, while others are oxidation and others are reduction. We need to separate them. All right? So, when we talk about light reactions and light-independent reactions, basically that's it. So that's the link, which was our first objective. Now, in the second objective, which comes in as the second section of this lesson, like we talked about, it is an extended lesson. So it means we have bridged one lesson and the other. So before we have this break to fall into the second part, remember, it's going to be a continuation. So even when you're going to be watching, don't move away, because we shall right away load into the second component of this, in which we are going to look at something like this when we say, how is carbon dioxide fixed? How do we get the glucose? What exactly are we looking at? Which we shall also wrap up with another clip as a continuation of what we looked at when we were beginning the lesson. The reason for merging these two together as a two-in-one is because they are interconnected and we still cannot separate them. So with this, as we see what next we are going to look at here, I want to break off from here and we return in a minute. Please don't go away. Thank you so much.